Dr. Brenyo, how do you take your coffee?
It used to be like coffee should only be coffee flavored coffee, and now it's more like, I'm a little more nitro cold brew, a little loosey goosey with it.
What's a hobby that you enjoy outside of work?
I'm a big carpenter. I make cabinets, and I like to do some more interesting projects as well. If you've ever seen the epoxy pour tables with inlay wood, I've made a few of those, and a couple kitchen tables. Last summer I cut the front stairs off my house, and I took them. They were straight and with a steep pitch. I changed the pitch and flared them out. So it's those kind of things that I enjoy doing.
Kind of switching gears, so why do you prefer MYNX central venous over other venous closure methods that you've tried? We
have gone through and worked with at least 4 different closure methods. The reason that the MYNX has worked very well and why I've stuck with it is it's very reproducible. It's allowed us to shorten bed rest with a lower failure rate than other closure devices. When I'm done with my procedure, my lab staff, my technologists, they, after going through and having a number of training devices where they actually keep a log, which we do for other things as well, they are capable of exchanging the sheaths and closing the vessels for me. So it allows me to do other things, going and rounding on patients, talking to families, and doing computer work. And that's different than other closure devices where it's much more hands-on on the physician side. So it improves my efficiency and the efficiency of the post-care for the patients.
What is your favorite feature of the MYNX central venous device?
Just the fact that it is very simple, straightforward, and with very little training you can figure it out quite quickly, and the fact that it's reliable and reproducible. If something had a high failure rate in various practitioner hands, that would be a concern. I like the fact that it's reproducible and very easy to use.
So for some of the more advanced techniques, like procedures such as LAA, how have you been able to adopt that into your MYNX central venous into those procedures?
We've developed a protocol that works very well for closure in that procedural space. It has allowed for us to do 6 or 7 watchmen in one room, have them recover and go home pretty efficiently thereafter. The failure rate is no different than if you were closing a smaller bore sheath, really. We used to use figure of 8, and patients had a fair amount of pain associated with the figure of 8, and it can be confusing for recovery staff to know what to do with the figure of 8 suture. If you have somebody who comes in part-time, they can be a little overwhelmed and unsure what to do, while closing the blood vessel and having a very set regimen for bed rest afterwards is really beneficial if there's staff turnover or you have somebody who's only in there part-time.
One other question I thought of, so you were part of our ReliaSeal study. Can you share kind of the impact of being a part of that clinical study and kind of what that experience was like?
So being part of the ReliaSeal study was honestly pretty fun. It was something different and out of the normal day-to-day. It was well received from a standpoint of the recovery staff, and with the larger bore sheaths that are required for various procedures, it was a good evolution, even within the trial, to get patients out, and that was where the staff really enjoyed it. I think the research process was honestly a lot of fun.